Alright everybody, so welcome to the Nexcelom Image-Based Cytometry and FCS Express 4 Flow Cytometry software for cell-based assays webinar. It's a long title, but today what we have for you is Amy Wilson from Nexcelom Bioscience is going to be presenting on the Nexcelom instruments, I believe particularly the CBA Vision, which allows you to export any of your Nexcelom cell-by-cell data to FCS Express 4 Flow Cytometry directly. And this is going to allow you to visualize any of that data that you've generated in a flow cytometry style using dot plots and histograms, and in a way that's very user-friendly in a kind of PowerPoint environment in which you can easily come in and learn FCS Express and generate very nice looking reports for yourself and for your colleagues and for publications. So without further ado, I'd like to introduce Amy Wilson from Nexcelom. She's going to speak to us for about the first half an hour here about everything you can do with the Nexcelom instrument. Then we're going to come back to myself to discuss how we can start working with that data in FCS Express 4. So let me turn over the presentation to Amy. And Amy, whenever you're ready.
Okay, I want to thank everyone for taking the time to dial in today. We're very excited now to have the Nexcelom Image Cytometry and our Vision CBA system combined with FCS Express software to really enable image cytometry for cell-based assays. So I'm going to give a little introduction to Nexcelom, introduce the Cellometer Image-Based Cytometry, and review several examples of some 20 microliter cell-based assays you can perform using our system. So we'll review cell cycle apoptosis, proliferation, GFP transfection, and autophagy. And before I hand this over to Sean, I'll go through a brief demonstration how we analyze an image and generate some cell cycle data using the FCS Express software platform. So Nexcelom was founded in 2003 in the greater Boston area. We're focused on automated cell analysis and committed to providing superior customer service and application support specifically for cell analysis and cell-based assays. We started in 2003 with a manual alternative to the hemocytometer, a disposable cell counting slide. And from there we went to an automated cell counter doing bright field only. And in the years since then, we've introduced fluorescence detection, both single channel and dual channel. And with the recent addition of FCS Express software, we can now do dual fluorescence assays with advanced data analysis to do cell cycle apoptosis and some other more complex cell-based assays. Cellometer is currently used by all top 10 pharmaceutical companies and also all 40 of the NCI comprehensive cancer centers. There are more than 1,000 cellometers out in the field being used. We've analyzed more than 1,600 different cell lines. So there's really a lot of traction and a lot of customer data showing the relevance and the quality of cellometer. So I'll take you through a little bit of the procedure. You start by adding 20 microliters of your stained cell sample into the counting chamber. We insert that chamber into the cellometer instrument. Then select our assay and enter our sample ID. And we click count. And within 30 seconds to 2 minutes, depending on the assay, we have the cell count, concentration, and diameter, and all of our data. We can view bright field and fluorescent images if we have a fluorescent model. And with the Vision CBA and FCS Express, we can click export and get our advanced data plots and histograms. Cellometer technology is based on an LED light source and a cooled CCD camera. There is no maintenance, no replacement of components. It's a very robust system. We utilize an imaging chamber, so there is no washing, there's no fluidics, no potential sample contamination, reduced potential for contact with any infectious samples, and this is really ideal for fragile cells such as hepatocytes and adipocytes. The optics module is integrated and user changeable. The Vision CBA includes 2 fluorescence optics modules Most customers will choose to look at the green channel and the red channel, but we have other options such as UV. This ranges from UV to far red. So why image-based cytometry? It's important to have accurate cell concentration and viability determination prior to flow analysis and cell culture experiments. It provides cell images for data verification that you might not get with a flow-based system. There's low volume 20 microliter testing for assays such as cell proliferation that allows you to actually do a time course study. And image-based systems are very accessible, easy to use, and easy to share. Very simple setup, predefined assays and instrument settings. There's minimal training, no maintenance. So it's very, very easy to use. It's very easy to share if you have a lab with multiple users. Specific examples of the power of Cellometer: you can remove debris from cell counts, visually confirm that only the cells of interest are being analyzed. In a flow-based system, cellular debris could be incorrectly counted. We can count individual cells within cell clusters. We can optimize parameters to identify the individual cells, view counted images directly on screen to ensure the accuracy of the cell counts before we generate our data reports. And we can also recount saved images using optimized parameters. So if you have a new user who's imaging, as long as their images are in focus, you can go back and pull up those images, adjust your settings a little bit, and recount. We count irregular-shaped cells. We have advanced algorithms in the software that can recognize irregular cell shapes. They don't have to be round. They can be multiple sizes. And we can also count cells based on size in a mixed population. This example is looking at mature dendritic cells cultured from PBMCs. So we have a variety of instruments for various applications. The Auto T4 is a basic cell counter that does bright field imaging and trypan blue viability, and that's for cell lines or cultured primary cells. We get very clean samples. We have the Auto 2000 for primary cell analysis. You can look at live and dead nucleated cells in a blood sample or a bone marrow sample using dual fluorescence. And with the Vision CBA, we can do more complex cells, such as adipocytes and hepatocytes, and also perform cell-based assays. So we've taken the dual fluorescence, added the power of the FCS Express software, a couple of hardware upgrades, and we allow customers to do cell cycle, apoptosis, autophagy, lots of other cell-based assays. This is an example of dual fluorescence of live and dead hepatocytes. And you can see on the image on the left, there's a lot of debris in the bright field image. And when you look at the fluorescent image, it's very clean. You can easily distinguish your live and your dead cells. So some of the fluorescent cell-based assays we validated include cell cycle, mitochondrial membrane potential, apoptosis using Annexin 5/50 PI, surface marker detection using antibodies labeled with FITC, PE, or other fluorescent tags, CFSE for cell proliferation, apoptosis assays involving caspase activity, GFP, RFP, YFP transfection, and other fluorescent proteins, agrosome detection, autophagy, and also ABC transporter assay. So I'm going to review some of the more popular cell-based assays. For cell cycle, we're looking at the different phases of the cell cycle. So in the G0, G1 phase, the cell is resting or growing. There's typically a fixed amount of DNA. If we look at cells in the G2M phase, where they're ready to undergo mitosis and divide, there is double the amount of DNA. And then cells in between in the F phase are in the process of duplicating their DNA for division. Our cell cycle assay calculates the percentage of cell population based on the fluorescence intensity. So in this experiment, we incubated cells with a cell cycle arresting drug, nicotazole. We incubated for 24 hours. We then fixed and stained the cells with rubidium iodide and analyzed them on the Vision CBA. So in the control sample, you can see that we have a very large G0, G1 peak. And as we add increasing amounts of nicotazole, the peak shifts over to the G2M phase. And we also did a comparison of these samples on flow cytometry and showed excellent correlation with about 10% of cells at the G2M phase before treatment in our control and 40% in the G2M phase following drug treatment. Apoptosis, or programmed cell death, is a natural process of self-destruction. It's a normal part of cell turnover and tissue homeostasis. Problems with this are linked to cancers, degenerative disease, autoimmune disease. Is a huge focus in clinical research. And necrosis is a form of cell death involving breakdown of cell membrane, more often caused by tissue damage. So to perform the Annexin V-FITC PI assay, we use two things. Annexin 5 has a high affinity for phospholipid phosphatidylserine, which is translocated to the extracellular membrane of the cell during apoptosis. And then tritium iodide dye can only enter and stain DNA in cells displaying a loss of membrane integrity, either due to necrosis or very late-stage apoptotic cells. So use of both Annexin V-FITC and PI enables calculation of percent normal, percent apoptotic, and the percent of compromised cells. So to perform this procedure, we take 40 microliters of our cell sample, add 5 microliters of Annexin V-FITC, and 5 microliters of PI, incubate for 15 minutes and wash. We then pipette just 20 microliters into a cellometer imaging chamber and analyze with the Vision CBA. And we're going to get automatic population analysis. We can look at normal cells, apoptotic cells, and necrotic and late-stage apoptotic cells. We have a colorized scatter plot, a linked data table, showing percent and concentration, and there's adjustable gaining, which I'll show a bit later, where you can move the gates and automatically the figures in your table will be updated. So the cell proliferation determines the number of cells that are growing in the absence or presence of certain agents. With flow cytometry, researchers typically take one endpoint reading due to sample limitations and high volume requirements, typically around 300 microliters for a large flow system. The 20 microliter volume for Vision CBA enables researchers to test multiple time points and also additional conditions using fewer cells. And the results of our collaboration are shown on this slide. We saw reduced fluorescence intensity indicating daughter cells and cell proliferation. Time course study shows increasing proliferation days from days 1 to days 3, 5, and 6. And the LSR2 flow results at day 6 match the day 6 cellometer results. And from this time course study, researchers could also see that at day 5, they're pretty much at the end of their experiment. They don't need to go to day 6 or day 7. They can really track the progression. For fluorescence protein expression, detect GFP, RFP, YFP, other fluorescent proteins, evaluating transfection using the user-changeable cellometer fluorescence optics modules. We can plot a control sample versus a treated sample and look at the GFP positive population. Cellometer gives you a concentration of cells and also a transfection rate. And in this comparison study, we got 93.4% on cellometer and 96% on a large flow system. So it's excellent correlation to flow cytometry. Autophagy is a cell-based assay that's really hot right now. We have a lot of customers asking about this when they come to our booth at trade shows. Autophagy is a normal process by which cells break down damaged cellular organelles and proteins to be replaced with new ones. And it's also a survival mechanism providing cells with energy in times of stress and starvation. It's a multi-step process that involves first the formation of autophagosomes, which are vesicles that capture and deliver the cytoplasmic material to be degraded. Formation of autolysosomes, where lysosomes fuse with the autophagosomes, and then the degradation of the cellular material. For this experiment, we utilized a CytoID green dye from Enzo, and that's used to stain both autophagosomes and autolysosomes and determine the concentration and percentage of autophagic cells in a population. As you can see the image here, the autophagosomes and autolysosomes will fluoresce inside the cells undergoing autophagy. And here we also did a time course study with rapamycin, an autophagy-inducing drug, and we looked at a time course and also different concentrations and saw the greatest amount of autophagy after 18 hours with 100 micromolar rapamycin. So just to recap some of the advantages of Vision CBA, you've got the small sample size, cell images for data verification, optimized for looking at cells and clumps and excluding debris. There's no risk of clogging when you're doing your analysis, fast user-friendly procedures, simple setup, user-changeable fluorescence modules. There's no washing, waste disposal, or maintenance, and minimal training. The system is easily shared by multiple users. And what I'd like to do now is just give you an example of how this analysis takes place. So typically we would insert a slide, we would focus, and then we'll click image. In this case, I have an existing image, and I'll show you how we would count. For basic viability, the counting is typically completed in less than 30 seconds. For some of the more advanced analysis, like for cell cycle or apoptosis, where we're doing fluorescence, it's typically about 60 seconds for the imaging and the cell counting. The nice thing about the Cellometer software is you can have lots of different assays in a drop-down box. You can have cell cycle, you can have different apoptosis assays. If you're doing a basic cell analysis for hepatocytes, adipocytes, or other cell types, you can set up individual assays for those specific cell types. And when you pull up your assay or your cell type, all the parameters for the instrument are already there, and the system just knows what to do to count the cells properly. Okay, and it looks from our progress bar that we're just about done counting here. All right. So this one, I'm just going to close this window, and we can take a look at our bright field cell image. We can take a look at our fluorescent image. We can zoom in and look at our cells. We can look at our counted cells. And we want to make sure that all of our single cells are circled in green, showing that they're counted. I want to make sure that some of our cell clumps are not counted and are circled in yellow. If this looks off, we can optimize our size parameters in the Cellometer software. And we can go back to our data, and we can export. And this is going to automatically open up the FCS Express software and pull up the layout that's preloaded in our software. Okay, now this is what our data is going to look like for a cell cycle. Temporary minor difficulty here. Okay, on the right-hand page, we have our cell population. And we can adjust this gate to either include or exclude some of our cellular debris. This will eliminate some of the debris in the lower left-hand quadrant. And this would include some of the debris. And then to the right, we have some of the cell clusters. Maybe we have doublets of cells that were not excluded in the Cellometer software, but we can exclude here in the FCS Express. And then on the left-hand page, we have our cell cycle histogram. We can adjust the gates. In this case, we're looking at the G2M phase. They're S phase gates, so we can move that. And as you can see at the bottom, here's our S phase at 4.3%. we were to adjust this gate, this percentage will automatically update. And there are lots of things we can do with this layout. You can change the color. We have it set up so that we have a legend and we have colored gates, but you can put in tags with percentages and labels. And Sean will go through some of the different things you can do with the FCS Express software to analyze the cell cycle data. So Sean, I'll pass this back over to you.
Yes, thank you for the wonderful presentation there. I was hoping that maybe you'd be able to show the export screen from the Nexcelom software one more time rather quickly, just so we can take a look at that. So what we have here is we click on the export button, and you'll notice here, if you don't mind going back to that real quickly, Amy, back one step. So you notice when we click on the export button, if you could click on that, you have this option at the top here to export data to Nexcelom data package, and then when done, open Nexcelom data package with FCS Express. So I just wanted to clarify that when you choose this option, FCS Express is going to be auto-launched from the particular analysis that you're doing within the Nexcelom Vision CBA. Just so everybody understands how that export process is taking place. So at this point, Amy has gone over quite a few things in regards to the Nexcelom software and their instrument. So I want to take some time and see if anybody had any questions about anything that Amy has presented before we go on to take another look at some of this data analysis in FCS Express. So if you have any questions, please find that questions box. You can ask your questions now. We'll just wait a few seconds here. If you have more questions that you come up with or you think of as we're going throughout the presentation, feel free to ask them here. We can certainly come back at the end. Great, and it looks like we don't have any questions right now, so that means Amy gave a very clear presentation, and thank you very much. So where Amy has left off, I'd like to pick up on. So I'm going to take the presentation back here. Alright, so where Amy has left off is where we're going to begin in FCS Express. You'll notice that when Amy auto-launched FCS Express, I'm going to open up that actual layout that she had open here, and I'm going to load that data in manually, and we'll just discuss this a little bit later. But you'll notice, again, one of the important things that with any of these Nexcelom-based templates that are going to be coming or become available for your use is that as you move these gates around, say this population gate, as we shrink it, as we move it, all of these histograms or other plots update in real time. And again, this is going to go for all these statistics down here as well. But what I'd like to do is come back to this, and I'm going to start here again from the beginning. And I wanted to give you just a quick background about FCS Express, since some of you that are coming from the image cytometry side of things may not know very much about FCS Express. But FCS Express has been developed as a powerful flow cytometry data analysis and presentation software, and we're very proud to be working with Nexcelom to export their image cytometry data directly into FCS Express for flow cytometry, so we can start looking at it in a flow cytometry style manner. The main principles of flow cytometry and image cytometry overlap, and these are such as taking multi-channel fluorescence data and plotting that data in scatter plots and histograms, so you can actually look for statistically significant differences in changes. And that's what FCS Express is going to do for you when you export that data to us, is we're going to be able to look at histograms, we're going to be able to look at scatter plots, we're going to be able to set up things called gates I'm going to touch on, and we're going to be able to do this very quickly and efficiently. And when you're done with your report or when you're done with your analysis, we can quickly and easily export all of this data to PDF files, to PowerPoint files, even to Excel documents and templates such as that. So we had showed you very quickly one of the reports that Nexcelom put puts together, or a template-based report. But what I want to start with today is to show you how we actually start from scratch in FCS Express. So how you can get to the point where you can create your own templates if you want, and we'll show you some of the templates that Nexcelom is offering, and we'll actually show you how you can modify these as well. So if you have certain modifications or formatting changes that you want to make that are particular to your publications or to your analysis or to your lab notebook, that way you'll certainly be able to come in here and make those changes. So the first thing about FCS Express, right now we're in FCS Express, is that it kind of shamelessly looks and feels and acts like Microsoft PowerPoint or Microsoft Office. I say shamelessly, and that's by design. That's because everybody has essentially used the Microsoft Office product, and if you have ever used Microsoft PowerPoint, you should not have a problem using using FCS Express. So when you want to open up a new layout or a new kind of template in FCS Express, we can do this by coming up here and clicking on the Applications button and New Layout. And I'll do this now. And you'll notice that we actually get a blank page that kind of looks like Microsoft Word, or it kind of looks like a slide in Microsoft PowerPoint. And anything that you put on this slide, whether it be plots or pictures or histograms, is going to be presented like a slide in Microsoft PowerPoint. And anything that's off the page will either be off the page or off the slide in Microsoft PowerPoint if you choose to save there. Or if you print this document, anything that's on the white section of the page will be printed, and anything that's off to the side here in the blue will not be printed along with the layout. But FCS Express operates in the ribbon and tab system, which is new for the Microsoft Office products. You'll notice that we can get to just about anything in the software by clicking through these tab buttons. We'll go through a few of these today, but just like Microsoft PowerPoint, if you want to insert something, you can insert shapes and text boxes and pictures just like PowerPoint. But for your analysis of your image data in a flow cytometry style manner, what you also are going to want to insert are things like histograms, dot plots, density plots, color dot plots. There's a whole array of different type of plots that you can choose from in FCS Express to do the type of analysis that you want. So if I wanted to start from scratch here, what we can do is let's start with a color dot plot for a particular experiment. And what I'm going to do is draw a box on the page just like you would draw a figure in Microsoft PowerPoint. And what I'm going to do is choose some data that was exported from the CBA Vision. In this case, I'm going to be looking at a apoptosis raw data, or apoptosis experiment. And within this experiment, I actually have two files here. I have a apoptosis necrosis negative control, and I have a treated control. You'll notice that in our files of type dropdown, we have a section here for Nexcelom files. So if you don't want to work in the template format and you'd like to work from scratch, you can simply just click on this to choose your .nextdat files, which is what's going to be exported from the CBA Vision. And then we can just click on the file you want to use to start with. And I'll click Open. And here we are. We have what we call a color dot plot. You'll see why we call that a color dot plot as opposed to a dot plot in just a few minutes. But what we have is a plot showing FL1 intensity and the size. We can change between any of the different parameters that the CBA Vision has put out just by clicking and holding on the axis. So you notice in this experiment we have a size parameter, we have an intensity parameter for fluorescence channel 1, and we have an intensity parameter for fluorescence channel 2. And if I let go of the mouse button while I'm on FL2 here, you'll notice that we changed this parameter. And again, I'll show you just by scrolling through a few of these different ones. So in this particular experiment, again, it's an apoptosis experiment, so we're going to be looking, I believe, at cells that are fluorescing in the FL2 channel. One thing about this data right now is that the scaling of the data has not yet been optimized. In the templates that you're using, that you're getting from Nexcelom, the scaling has been optimized already. But since we are beginning from scratch here, sometimes you have to make some scaling changes. And to do that, or to make any sort of change to any plot in FCS Express, you can right-click on that plot and choose Format. And by right-clicking and choosing Format on a plot, you have access to all of the formatting options in FCS Express. It's quite an exhaustive list. This is going to work just like in PowerPoint if you want to format, say, a figure name, if you want to format the border or the size. But there's also things in here relative to plots themselves, like color dot options and axis options. So by coming into the axis options, what I'm going to do is actually change this y-axis scale to log with negative, and I'll change the x-axis scale to log with negative. And when I do this, I can click the preview button. And when I click preview, you notice how these populations actually pulled apart now. When we view these in a log scale, it allows some negative values to actually display some of the lower numbers here. This is much better suited for this particular data set, and it's going to bring out our populations of interest quite quickly and easily. So I'm going to click OK to apply this change. And now we're working with the correct type of scaling for this particular plot. Now, we can certainly do this for histograms as well. If you want to view either one of these channels in a histogram, you can simply click on Histogram, draw a box for that histogram, change to the parameter of interest. In this case, it's going to be FL2 intensity. And again, you'll notice the scaling looks a little strange because we're actually scaling linearly here. But again, if I just do a quick right-click and format and choose my axis and change to log with negative and click OK, we have formatted this plot, and now we're looking at a better scaling of this data. Now you may say this histogram still doesn't look very pretty. The great thing about FCS Express is that you can format any of these plots any way you like. So if we wanted to actually smooth this histogram data, again, we can right-click and format. This is going to be in our overlays, and we can smooth this data a little bit. We'll change it to 7 and click preview. And you notice that the histogram now looks a lot nicer than it did before. Keep in mind that smoothing is not going to change the values of the data. It's only going to be changing the display of this data itself. So what we want to start doing with this particular experiment is essentially we know that this population in the upper right is going to be our apoptotic cells. And what we want to do is figure out how many are there, what percentage of the entire image that these cells come from. Keep in mind that each individual dot in this dot plot represents a cell from the image that was generated by the Nexcelom Vision CBA. And to deal with this type of thing, looking at multi-parametric data and trying to figure out what populations are what we do in flow cytometry is something called gating. For everybody, if people are familiar with flow cytometry, everybody knows what gating is. But for those of you who come from more of an imaging background, gating is actually something akin to creating thresholds for your data. So in the imaging world, we would say if we want to look at the cells that are positive for FL2, we would come in and create a marker and look at only the cells that are within this region of the threshold. Essentially, a gate is a two-dimensional threshold where we're actually looking at a value going in the y-axis here and a value going in the x-axis direction. So to insert a gate, we can come over to our gating tab. We can choose any of the different gate types that we have. In this case, I'll choose a rectangle, and then we can just draw that gate somewhere on the plot. We can name that gate anything we want, so we'll call this apo cells, and we can give that gate a color and keep it red. And now any of the cells that appear within this apo cells gate are appearing in red. That is why this is called a color dot plot, because we can view the cells in a certain color when they fall within a certain gate within that plot. Now, we have this whole other population down here. Maybe we're still interested in this population. So if we want to create another gate, a polygon gate, where I can actually click and grab a few of these things, we'll call these live cells. And now they're going to be appearing in blue. Now, in this particular experiment, forgive me as I'm not too familiar with the staining patterns, but something we do in flow cytometry is that we do what we call hierarchical gating, where we can take gates and make them children of each other. And let me insert a gate view from the Insert tab Gate View to show you what I mean. In this particular experiment, we've set up two gates, the apo cells gate and the live cells gate. But what if in this particular experiment, all we wanted to do is look at the cells that fall within this particular gate to begin with? Maybe we're not interested in the cells that are fluorescing very low. We can call these interesting cells. And if we'd like to only look at these cells, what we can do is take this interesting cells gate and we can drag it and drop it directly onto the plot. And you notice that that excludes everything outside of this interesting cells gate. And now if we only want to get statistics for these live cells here that are interesting and the apoptotic cells that are interesting, what we can do is drag these in the gate view underneath the interesting cells gate. And now you'll see that there's actually a branch here. And this means that these live cells are only cells that are interesting, and these apoptotic cells are only cells that are interesting. So this is a way in flow cytometry that we can look at subpopulations of cells or look at cells in a multi-parametric way. So another thing we could do with this particular assay is say, let's start gating some cells by size. So what we can do is maybe we want to create a gate here and only choose the largest cells to look at. So if I call this large cells and we'll make this gate pink, you notice that we're only choosing the large cells. They fall in this upper part of the y-axis here. And we'll drag this over. If we wanted to look at only live cells and apoptotic cells that are large, we can simply drag these under here, apply the large cells gate back to this plot, and we can flip back to FL2. So now in this plot, we're only looking at cells that are large, and we're finding out how many live and how many apoptotic cells there are within that particular population. So FCS Express is going to allow you to quickly and easily create these gates and these plots and get into this multi-parametric data very quickly. And that goes for applying gates back and forth. So if I only wanted to look at the large cells within this histogram, I could simply drag this large cells gate onto the histogram, and now we know that all the cells appearing in this histogram are only large. And as I came back to my size here, if I change this gate, you'll notice that our histogram is updating for us in real time. So this is a great way to zoom in on populations of interest and see what's going on in channels that are different than the channels that you're regularly looking at. So once you have some data like this, the next thing that you probably want to do is get some statistics out of this data. We make this very easy to do in FCS Express. We can do this by right-clicking on a plot and choosing Gate Statistics if you're working with a two-dimensional plot. And you'll notice that we have an entire default statistics table that pops up. It's telling us, in this case, how many cells there are in the entire image or experiment, how many of them are interesting, how many are large, how many are live, how many are apoptotic. We're also getting intensity values from these, X and Y geometric means, and we're also getting percentages of these cells as well. So we know how much of the gated population or how much of all of the cells that these particular cells or gates represent. We can certainly do this for histograms as well by right-clicking on the histogram and choosing statistics. We'll get all that information, including things like the high bound and low bound of this histogram, CV values, peak channels. And if you don't want to use these default statistics tables here, we can certainly set up any type of custom statistics that you want in the software or custom views of these, just like you saw in some of the Nexcelom templates. We can do this by inserting a text box. And I'm going to insert that text box. I'll call this My Data. So we can just type in there. And in this My Data text box, we can start working with something in FCS Express that are called tokens. And tokens are live updating bits of information or data that come from these plots. So if I wanted to insert a token for the percentage of live cells in this gate, I can just drag this box into the text box, release it, and we'll choose the number of events. And when we click OK, we now have a value in gray, and gray indicates that something is a token, and we'll type in number of events here. And these tokens, anywhere they are within the software, as you change a gate, you'll notice that that value is going to update for us. And if dragging gates in is not your thing, and you maybe want to just grab this number of events or apo cells from this table, you can quickly and easily just drag that in here as well. And now we have number of events, and we'll call these apo cells, and we're getting that value of 450 for that particular plot. And again, this will update for you in real time. So that's how we can start building up some very nice templates and some very nice reports in FCS Express to start looking at all of your Nexcelom data. The other thing I want to mention, I'm going to delete some of these text boxes here, is how we can actually export this data very quickly in a way that we can go out to, say, PowerPoint or PDF. And I also want to show you how we can work with multiple files at the same time. Because when you're doing these experiments, you're never just going to have a negative control, right? You're going to have a negative control, a positive control, and however many samples you have in between. So to show you how we work with multiple data files, what we can do is come up here to our Data tab in the data list. And I'm going to change this data list real quickly so we're only looking at file names here. But you'll notice when I loaded this first data, this negative control, the other experiment that we had in there, the other experimental control, was the treated cells. Since they were within the same folder, when we loaded up this layout, they came into FCS Express together. And now that we're in the Data tab, you'll notice that we have this Change Data on All Objects button. And what this button is going to allow us to do is change between all the different files in the data list. So in this case, we only have 2 files, but if you had 100 samples in between your positive and negative control, you can hit the Next button and watch as everything in this layout changes for you. So we're changing to the treated control. All of our statistics have updated. Our histogram has updated. Whatever statistics or gates that we had here in this report would update rather quickly and easily using these next and previous buttons. But if maybe you wanted to do something like create a histogram overlay and actually look at these 2 files on top of each other, this is something we can do very quickly, easily. So if we have the negative control down here in our histogram, and we wanted to view the treated control on top of that, we can just drag that file on top of the histogram, release, say add the file as a new plot, and you notice that that new file is now indicated on this histogram as an overlay. And what I'd like to show you real quickly is we can right-click and choose Format, and we can make these overlays look very nice. In our overlays, you now see that we have the negative and the treated control, but we want to change our smoothing on the treated control to be up to about 7 to match the other one. Let's say we want to fill this histogram area as well. We click Preview, and now we have that area filled as well in that histogram. Also notice that we can apply different gates here to each of the overlays. So maybe I only want to look at the apoptotic cells in this particular overlay. Now we're only looking at the cells for the second file or the treated control that are considered apoptotic. We can see these side by side. And we also have some other options here to view these as stacked on top of each other. And the other thing I did not show is that we could view these histograms in 3 dimensions as well. But if this is all you want to do with your analysis, this is your analysis from scratch, and we want to say export this data very quickly so you can actually show this data to your PI, to your publisher. You can put this in your lab notebook. All we have to do is come up here to the Applications button and say Save Layout As. And we have many options. We can export this as a picture, as a PDF file, but what I really like is exporting to PowerPoint. So I can say save it as a PowerPoint presentation. We'll call this a test on my desktop, and we'll open it after we save. So when we click the Save button now, what happens is FCS Express is opening PowerPoint here, and it actually just saved this data to PowerPoint. And if I zoom in here, you'll notice that we have the plot in PowerPoint. We can move this around. These are super high resolution plots. We can make these as big or as small as you want. So if you want to put this data in a poster, this is a very quick and efficient way to get that in there. And you can even, use these as a figure in a paper if you like. But notice that all the statistics are coming in as well. So that being said, I think that's a lot of the basics of FCS Express. I just gave you a lot of information in a very short period of time, but what you are going to see at the end of this webinar is that we have many resources to help you get started in working with these layouts yourself, if there's any changes that you've made. But until that point, we'll come back to where those resources are located at the end. What I want to do is spend a few minutes talking about some of the other templates and experiments that Nexcelom provides. And let's actually go back to the cell cycle analysis template that Amy had provided us earlier and take a look at what's going on here. So in this particular layout, as we mentioned, we have a gate view in here where we can actually view all the different gates associated with this layout. And any of these gates, if I wanted to drag them onto the histogram, we can certainly do that and see where maybe only the G2 cells lie. And again, as we update this marker, that G2 only gate is going to update for us as well. Anytime if you want to remove a gate, you can certainly come up here to the gating tab and go back to no gate to change that. And we can make any sort of change in the software as well. This is a good time to tell you about the undo button. The undo button is located up here in the upper left. You'll notice that it says undo the resize. And now if I wanted to undo a few of the gating changes I made to get back to where exactly we started, you can see that we can go all the way back to the beginning. We say unlimited undo. It goes back to 30 steps. If you've made more than 30 mistakes, you may want to rethink what's going on with the analysis anyway. So keep in mind that that undo button is there. It's very useful for you as well. Again, in the statistics table that Nexcelom has set up for you guys, we have all of these values appearing here as tokens. Again, remember, these are all grayed out and they're represented as the percentage of gated cells. Anytime you can click on these tokens, you can double click. If you wanted to change the statistic, instead of using the percentage of gated cells, maybe you wanted to look at number of events instead. We can certainly choose number of events. And now in this case, in this one particular column, we're looking at number of events. So you can quickly and easily come in here and change any of these things from these default templates that have been set up. Now, I also, when I mentioned tokens, I mentioned that we can pull information from files themselves. So in this particular experiment, you'll notice that these values are all grayed out. The instrument, the OP6 modules, the assay names. These values are actually being stored as tokens that we call keywords. So if I were to right-click here and choose Insert Token and choose a keyword, what we can do is come to our list of keywords here, and we can choose any of the information that was stored at the time that this image was taken or this image was analyzed. So if I wanted to come in here and I wanted to put in my optics for this, again, we can simply choose optics and click OK and click OK. And here's all of our optics settings for when we use that experiment. And you remember earlier I showed using the data list to switch between files, next and previous. If you were to do that, in this case, all of these values would update as well, the date of the experiment, if there was changes in the exposure time or anything, all of those changes would occur for you rather quickly as well. The other thing I wanted to show you about this particular cell cycle model, one way that we can actually start maybe looking at the different populations as colored populations is to create an overlay here where we color the cells that fall within a gate a certain color. And let me show you what that means. If I wanted to come over here to our second page and drag and drop this plot on top, what I can do is say add the overlay, add the file to a plot as a new overlay. And maybe this doesn't make a lot of sense, right? It's actually the same file. But what I can do is come into the Format tab here And in my overlays, I can choose the second overlay. And I can say, let's only look at the G2M population. And let's color this green because that particular gate is colored green here. And let's fill that area. And when we click OK, you notice that the G2M population is now filled in green, and that's going to update for us as we move this around. So once you've done this, you can actually do this with whatever populations you have in this histogram, and you can really create some nice modifications to this report. That way you can see which cells are being counted in the analysis where. That being said, FCS Express also has some options for cell cycle that go kind of above and beyond just this static type of modeling here by setting up markers and gates. We can actually mathematically model your cell cycle data using a Flow Cytometry mathematical modeling piece of software that's built directly into FCS Express. That plugin or that build-in is called Multi-Cycle. You can actually come to our Insert tab here and choose Multi-Cycle DNA. So if you're working with the cell cycle data and you have a good cell cycle looking peak, we can insert a multi-cycle DNA plot here. We can choose FL1, this is the particular parameter we're interested in, and click OK. And what this is going to do is create a multi-cycle plot, or a plot that's based on the theory that DNA that is dividing, or that's in G2, has twice as much DNA as something in G1. And this type of modeling is going to allow you to quickly come in here and right-click and choose cell cycle statistics, generate things like the number of cells or the means for G1, the percentages, all the CV values automatically. We can also get things like DNA model statistics. So with multi-cycle, you notice in the tab here, there are actually many models that we can choose from. Typically the one with the lowest chi-squared value is what you'd like to choose. But we can also get things like DNA experiment statistics. So it's saying that there's no abnormal DNA. It's telling us how much is in the S and G2 phase, what are S phase confidences, and also some of the statistics associated with all of this. So the basic model of cell cycle is included free with FCS Express. So if you have FCS Express Flow Cytometry provided by Nexcelom or that you've just purchased yourself, you will have this available for you to try. So it is a very interesting way that we can apply some of our flow cytometry style analysis that has been built into the software for many years with this image cytometry data. And these multi-cycle plots or these DNA analysis plots work quite quickly and easily. So I wanted to leave you with one more assay here. This is an Apoptosis Annexin assay, and you'll notice that in this assay, again, we're looking at FL1 versus FL2, but we've set up this color dot plot, and we've set up all of our colors in here a little bit more nicely this time, and we've used quadrants. And as we move these quadrants around, you'll notice that the colors on the right here for these quadrants or these gates are updating for us in real time. That's because we've actually done what we call linking here, where we link these quadrants to a gate, and that allows us to dynamically update the colors that you're viewing these cells in, and along with all the statistics down here for gated cell percentages. So this is again a great starting point. If this is all the data that you're interested in and you want to get this very quickly and easily, you can simply come in here and load your data into here. If you have more experiments, remember that you can keep these in by putting these into your data list here. Put as many as you want, and then you can analyze these simply by clicking the next and previous buttons. What we don't have time to cover today is actually batch processing. So if you did have 100 samples here, we could use these batch options over here to actually analyze all of these 100 samples automatically in an iterative fashion, meaning that we can say choose save to PowerPoint, and we could choose save to Excel, save to PDF, print. And if you had 100 items or 100 data files in this data list, you click Run on this batch process, it will do all of these processes, PowerPoint, Excel, PDF, all at the same time, and then within just a few minutes you'll have PowerPoint reports for everything in your data list, making your life and the life of your researchers in your lab much easier and more efficient. So we're kind of coming to the end of the webinar here. I mentioned these two applications templates. Of course, Nexcelom has many, many applications that they can use in the CBA software. On the Novo Software website, and I believe Nexcelom has this on their website as well somewhere, is that we have some example application layouts. So if you wanted to actually come in here and download these to take a look at what's going on, you can just come up here and search the Novo Software website for Nexcelom and find these. And we actually have example applications of each of these, showing you assay information, showing you a PDF. If you click on any of these images, it will give you a nice view of what that report looks like. So you can get down here into any of the assays that they currently have out right now, see what's going on with these, and you can certainly download these layouts and load your data into them as well. But what I'd like to finish up with today, before we open up the floor for questions and answers, is I'd like to kind of summarize what you saw with FCS Express today. Essentially, the FCS Express Flow is going to leverage the most powerful and proven tools of flow cytometry software with new features to help improve your Nexcelom cytometry analysis. Things as multi-parametric gating, using histograms and overlays. And as you saw, a lot of the drag and drop things, like dragging and dropping overlays and gates, make the analysis very quick and easy. Everything is state-of-the-art with the visualization, plotting, graphing, data analysis, and it's all in one package here for you. FCS Express also has a free reader and online collaboration tools. So this is a way that you can collaborate with your colleagues. Not everybody has to own FCS Express to open up a layout. You have to own FCS Express to actually save, make changes to that layout and things like that. Just like with a PDF, not everybody owns a PDF writer, but everybody can open up a PDF file to at least take a look at it. That's what our reader will offer you. But again, it's very intuitive with a Microsoft PowerPoint-like feel. So yourself and your colleagues should be able to open this up, especially with the templates that are pre-designed. You can come in and get started quite easily. And in the end, Novo Software is going to provide many resources to our customers. We have tutorials for working with flow cytometry, for working with the Nexcelom data. We have sample data, we have an online forum, and we have a very good customer support system. So if you have any questions about working with your data in FCS Express, you can contact us at support@Novo Software. We'd be happy to answer those questions for you, no matter what they are. And if you currently don't have FCS Express, but you do have the Nexcelom CBA Vision, and you want to start opening up those Nexstat files, or even just working with FCS Express to get an idea of what it has to offer, you can get a free trial from our website at denovosoftware.com. You can go here to our website in the download section, FCS Express demo, to get that free download for yourself. That's going to include working with Nexcelom data files, the multi-cycle, everything that's in there. We also have some other webinars going on this week. If anybody is interested in some more image cytometry based things, you can come here to our website to take a look at what we're doing with working with cell profiler segmentation and working with image-based segmentation as well. These are some ways, the segmentation, outside segmentation, are ways that you may want to start thinking about for some other applications outside your Nexcelom device, and you may find these interesting to stop in and take a look at. So all that being said, I'd like to open up the floor to questions. Are there any questions that anybody has for things that we've gone over today? Is there any questions for the team at Novo Software or the team at Nexcelom? It looks like we had a few questions that came in that were answered. Okay, are there any questions for myself or Amy? One question is there a possibility of providing an on-site demo? We can Novo Software is happy to do a lot of on-site demos. We typically visit labs all across the country and all around the world. So you can contact us at support@Novo Software if you'd like to get more information about having us come out to demo FCS Express. And we also have the contact information here for Nexcelom as well. Amy, would you like to comment on doing any on-site demos as well? Is that something that
Sure, we can certainly send out our application specialists. They're in the field and they do demos of the Vision CBA free of charge. They could just do basic cell analysis, or they can bring in a cell cycle demo kit or an apoptosis demo kit and show you firsthand in your laboratory how it works.
Great. And another question that we got is, can we do compensation for different fluorochromes? For those of you in the imaging world, you may not be as familiar with compensation. Compensation is a way in flow cytometry that we can start pulling apart fluorescent channels that essentially overlap in the light spectrum to pull apart the populations using negative and positive controls of known negative and positive fluorescence. This is something we've been doing in FCS Express for many years. We can certainly do this in the current version. We can do it with Nexcelom data if that's needed. Compensation probably will not be necessary working with the Nexcelom data because their filter sets are set up so that there are not overlapping channels. Typically most microscopes work like that. But if there ever is a reason that comes up, you can certainly try it out and see how that works for you. So are there any other questions for Amy or myself? Great, and so today I'd like to thank Amy Wilson and the team at Nexcelom for coming out and presenting this webinar with us. It's always a lot of fun for us to come online and work with our partners and to work with the customer bases from both worlds. So we really appreciate that time and effort that Nexcelom took to come out today. And with that, I'd like to end the webinar. And if anybody has any questions after the fact, feel free to reach us at any of the email addresses that you see up here on the screen right now, or at support@denovosoftware.com. So Amy, is there anything you wanted to add before we end today?
No, just thank you everyone for coming. If you have any questions, you want any more information, just feel free to visit our website. We do have links there where you can request a demonstration or download any information that you need.
Great, and this is what the Nexcelom website looks like. Again, that's www.nexcelom.com. You can go there to get any of that information. So thanks again, everybody, for coming out today, and we hope to hear from you soon. Have a great day.